Right, MHC. So MHC stands for Major Histocompatibility Complex, right? And it's fancy, fancy stuff there. Okay, so what it is, all MHC is either an arm holding up antigen. So we mentioned about how that antigen could be red top marker, right? So it's an arm holding up antigen that either got from outside of the cell or from inside of the cell. Any cell that has a nucleus can get infected inside. And so if they pull something from inside that didn't belong there, they'll hold it up on MHC1. So that's the arm that says, this is MHC1 that is holding out something I found in here. Come take a look at it. So that arm is just a thing there. That MHC is just the arm that's holding the thing. It doesn't hold a specific antigen. It can hold any antigen that it found inside, okay? MHC2. MHC2 is an arm that's only found on antigen-presenting cells. And MHC2 is the arm that antigen-presenting cells put up saying, I picked this up from outside of the cells. And so if I'm a phagocyte, like a macrophage, or if I'm a B cell that's sitting in the lymph nodes scanning everything coming through, those guys can pick up antigen from outside and then say on their MHC2 molecule, I found an antigen. Come and check it out for me. Okay, so that's what MHC is. And those are what the two versions are. MHC1 will display things that came from inside. MHC2 displays things from outside. All right, so how, once we have something displayed on MHC, or if we're interacting with antigen specifically like a B cell does, how are they going to recognize specific antigen? Because MHC can present any antigen at all the 8.6 billion different antigens. But how is your B cells or your T cells going to interact with a specific antigen? And that is through their receptors. So B cells and T cells have special receptors that are specific to a specific antigen. All right. So your B cell has a receptor that looks like this. Does this look familiar to you guys? It probably does. It looks an awful lot like when we learned about antibody, right? And so as it turns out, B cells essentially use antibody as a receptor on the surface of their cells. Antibody itself is just free floating protein version of this. But before our B cells can make that free floating protein version of this, they have to use it as a receptor on the surface of the cell to let them know they've actually found the antigen they've been looking for their whole lives. So it serves as a receptor. Receptors are always molecules on the surfaces of cells that basically signal to the cell that they've interacted with something. Okay, so on B cells, that's how they're going to be recognizing things. On T cells, they have their own receptor, and it actually works in a very similar way. But with B cells, when they activate their receptor, known as an immunoglobulin, we abbreviate as IG, that's immunoglobulin, and this particular receptor is called IgD. The immunoglobulin that serves as a receptor on the surface of B cells, aka IgD. We'll have a whole bunch of other IGs. There's 4 other ones we need to know, but they're all made in response to having been activated because IgD found something first. So IgD is our receptor. And then when that B cell gets activated, it'll be able to make the antibodies in whatever versions that we need to make later on appropriate to the response. And those will be secreted out of the cell as antibody, not attached to the surface of the cell. Okay. The T cell receptor is always attached, we'll say on surface of the T cells. It doesn't secrete it. It doesn't put its thing out there to interact with things. And so because B cells, their whole purpose of a B cell is to make antibody. That's what it does. First, it'll wait out in the lymph node. Its IgD receptor will recognize that, hey, I've picked up red top marker. Then it will get activated. And then when it is activated, it will turn into a bunch of B cells. Some of them will be memory cells. Some of them will be regulator cells. And some will be effector cells. And the effector ones are the ones that are doing the job. And what is the job of effector B cells? Make antibody. They're also known as plasma cells when they do that. And so when they're making antibody, those effector cells are cranking out soluble versions of just that protein, that immunoglobulin. And they're going to have their own jobs, which we're going to talk about in a minute. But the B cells aren't doing anything with the pathogen. The antibodies they make are interacting with the pathogen, right? So this is just proteins that they're making that are interacting with the pathogen, and they're going to have a job in containing the pathogen and the infection and everything like that. That's the proteins that they make. The antibodies do that. But T cells have a different function. T cells have two major groups, the T helper cell and the cytotoxic T cell. I'm going to talk about them and their functions in a minute, but their cells are actually doing the work. The helper T cells are the captains. They tell everybody that they can go ahead and do things. The cytotoxic T cell is going out there and killing all of your infected cells. And so those guys have their jobs as cells. They're actually doing the work. So we call T cell mediated responses, we call that cell mediated responses. With antibody-based response, we call that humoral response because it's in your humors, it's in the fluid of your body. So it's found in your plasma. We've got these antibodies all flowing around, and the antibodies are doing the work, not the B cells. So humoral response versus the cell mediated response. Okay, so our B cell receptors, those antibodies that we looked at, let me get to the picture part. Here's our nice happy little antibody over here. We've already seen that before. This part of the antibody, these long parts here, the inside of the Y, these are called the heavy chain. These little side pieces out to the side, they are called, these guys are called the light chain. All right. The top part of the Y part of it is the part that interacts with antigen, And so we have a lot of variability from B cell to B cell, or from antibody to antibody made from the different B cells, with a lot of variability in that region. And so therefore, go figure, we call it the variable region. There's a lot of variety in there from B cell to B cell, okay? Then we have the handle of the Y, the bottom part of the Y. We call this the constant region. So the constant region of the antibody dictates its functions and where it can be in the body. So function and location. So this part doesn't change a lot. This is basically only going to change basically in order to change it from IgD to the other forms of antibody, whether that's IgM, IgG, IgA, or IgE. So IgD is the receptor form, and then once we get activated, we'll start cranking out antibody, and that'll be one of those other forms. It just depends on what signals we're getting and what stage we're at in the response. Okay. So when we are actually looking at these at the molecular structure level and looking at why we determined to call it a Y shape in all of this, this variable region is also known as the FAB. I think of this as like what makes the antibody and its function, so FAB. As it turns out, it's FAB and FC. So A, B, and C here. And the C, constant. So the two parts of antibody, the FAB portion is the variable region, and the FC portion is the constant region. I know there's lots of terms for these. I didn't make up these terms, but you got to learn them, okay? So there's our terms for what antibody is and what these structures are. There's another slide that's dedicated to the FAB and the FC, but all I need you to get from it is that the top variable region is called the FAB and the bottom constant region is called the FC. I'm not going to get into any more detail than that. Okay, so that's our antibodies. T cell receptors, they also have a variable end where they're interacting with their antigen, but they're always going to be attached to the membrane. So they don't need like different versions of a constant region. They just have the region that's attached to the membrane. And there's always going to be in the membrane. It has this antigen binding site at the top, just like the Y-shaped antibody does, but we're never going to secrete it out of the cell. How do we get all of the variation to have all of these billions, like 3 billion or whatever it is, ability to see all of these different billions of different antigens when we only have 20,000 genes? I have a little video for that that explains it a little bit better than my words can explain it. But it basically takes different parts, segments of your genes, and picks and chooses from each category, matches them together, adds in or deletes nucleotides, and then just gives a lot of variety from that. So we're going to play the video for that because it explains it better than I do. and it's got pictures. Hopefully it's not too janky. Wait for it to load. There you go now. There it goes. Jeez. It is so slow.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. Billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.
Okay, so I feel like that's a pretty cool thing that our bodies have developed in order to fight off any potential pathogen. And this is something that occurs not just in humans, but in any mammals in general. They all have similar systems as this one in order to combat potential diseases. Okay, so if you have a B cell that only sees COVID spike protein, you only have one of them that would only see COVID spike protein. How do we get all these B cells that are making all this antibody in response to something? And this is going to happen through our clonal selection process. Essentially, what will happen is with your gene reassortments and your ability to, target any antigen in the whole universe, that includes cells that can target self molecules. And so we don't want those guys to stick around. So early on in the development process of these guys, we will weed out any cells that definitely attack self. And so at least that's the goal, right? And so we're trying to train those cells to react to self. And if they have any reaction with self, we'll just kill them. They aren't allowed to go on further than that. And of course, this is not a perfect process, unfortunately. But we try to get rid of as many of the anti-self clones that we can. This process, getting rid of anti-self clones, is called clonal deletion. And this leads to immune tolerance. That means our immune system tolerating self. The next step of that means that we have this whole repertoire, as it says, a repertoire or group of all of these cells that can interact with non-self things. We have one for COVID antigen, one for HIV, one for flu, and et cetera, and et cetera, and et cetera. And we have these billions that are just waiting to see COVID spike protein and et cetera, et cetera, et cetera. Well, when COVID spike protein shows up, in this case, red triangles, okay? When red triangles show up, the guy that reacts to red triangle will clone itself out and proliferate first. Will make copies of itself. And then after that proliferation happens, we make all of our copies, then we will differentiate. All lymphocytes do this. So all lymphocytes, when they see their antigen, they will proliferate first. Not like, oh, we've seen our antigen, now I'll make one, be a memory cell and one be this. No, first we make a whole load of copies, and then we segregate this set of the copies for memory, regulatory cells, so that we don't overreact. And these guys will control and make sure we're reacting enough or not too much. That's the point anyways. Regulator cells or effector. And the effector ones are ones that are actually going to do the job during the actual infection. Okay, so these are either our B cells that are actually making the antibody, aka plasma cells, or they are the T helper cells that are controlling as captains, or they're the cytotoxic T cells going out and killing infected cells. All right, so we already talked about immune tolerance. Again, B cells, they are going to do all of their maturation in the bone marrow. And then once they have matured in the bone marrow, that one cell that can potentially see COVID spike protein is just going to hang out in a lymph node or in lymphatic tissue. So B cells start in the bone marrow. This bone marrow, okay? Not something else. All right, so it starts in the bone marrow, and then they will move to reside in the lymph tissues. This is the lymph nodes or those salt malt galt guys, the associated lymphoid tissue. They will wait out there and just filter out as things are going through. The T cells, they start in the bone marrow. They mature in the thymus. And then once they've actually matured and they're ready to go see COVID spike protein or whatever, and they've gone through all of the growing up that they need to do, and they're ready to actually go out there to wait for the war to start, these guys will actually circulate. So they'll either be actually in your bloodstream circulating, or they can move in through the lymphatic tissues as well, but they can kind of move wherever they need to. So it says down here, it's estimated that 1 billion T cells pass between the lymphatic and the general circulations per day. That's pretty impressive. So you're constantly circulating all of your T cells throughout your body, from your lymph and in through your circulatory system and back, just constantly. These guys are constantly surveying all the time. So this is going to bring us to our topic of how these things are going to actually act in your immune system. And it is easier if I draw out the process rather than just show you blocks of words. Okay, so we're going to start with the start. As it turns out, none of your lymphatic cells, your B cells or your T cells, can actually do their job without the go-ahead from their captain. So first things first, once we have an antigen-presenting cell plus the appropriate antigen, these guys will activate a T helper cell. All right, so these guys have a special marker on them called CD4. And honestly, there's 2 CDs that I need you to worry about, okay? There is, come on, thingy. Just go away, nobody cares about you. Okay, there's 2 markers I need you to worry about, CD4 and CD8. I feel like a 4 looks a lot like an H, okay? And you'll see why I'm going to make that correlation in a minute. A 4 looks like an H without one of the legs. So that's how I remember that the 4 goes with the helper, okay? Once the CD4 helper cell is activated, it will turn into 2 versions of itself, a T helper 1 or a T helper 2. The T helper 1, this guy regulates inside stuff. The TH2 regulates outside stuff. This, thank goodness, correlates with our MHCs also. So your MHC arm that's holding up stuff from inside correlates with your TH1 helpers that are going to regulate the inside stuff. Thank goodness, at least that stuff is going to correlate. So our T helper 1 or our T helper 2. So this is the gist of our captains. All right, so we're going to keep this guy over here on the side so we can refer to them in a minute. So how does this stuff actually happen? You have crap that got into your body because you breathed it in or you stepped on a nail. The crap got into your body somehow, right? And in that area, we will have your injured cells calling out for help. And because they're calling out for help with their chemical signals, we have now called in through chemotaxis our first main line phagocytes, the neutrophils, right? And the neutrophils are going to eat stuff up and just either poop it out or die or something. So the neutrophils are in there eating up stuff, but they're not doing anything with it outside of that. But they're also sending out signals saying, hey, I keep eating up stuff. We're going to need more reinforcement here. So they're sending signals too. And so what moves into the area to help clean up that mess? The antigen presenting cell, in this case, a macrophage. We'll abbreviate it like this. So this is a macrophage. So the macrophage comes in, it eats up what the neutrophils left behind, as well as the neutrophils that were there, because they did their job and they're done. So when the macrophage comes in and eats up all that stuff, they picked stuff up from the outside. So where are they going to present it on? MHC2. This is outside stuff, right? All right, so they'll put that antigen on MHC2. And what happens is we have a special receptor on our T cell called the T cell receptor that recognizes that specific antigen being presented by the MHC molecule. So MHC can hold up any antigen, but once it is holding up that antigen and the specific T cell receptor that recognizes that antigen, that's when this binding will occur. So now we're presenting COVID spike protein, and the T cell that can recognize COVID spike protein has come up and interacted with its antigen. It's been waiting its whole life to see. And this just isn't any old regular old T cell. No, this is a T cell that has a special marker on it. If we're talking about outside stuff, this is going to be CD4. As it turns out, CD4 only interacts with MHC2. Only interacts with that. So CD4 only sees stuff presented on MHC2. This is key because your captains are going to regulate both sides of the response, but your captains only get activated by outside stuff. You can't react to inside stuff until you get signals from the captain that got activated by outside stuff. But 100% of the time, stuff didn't get into your cells magically, right? It had to get in there from the outside. So if you have an inside infection, it had to get there from the outside. So you will always have both if you have an inside infection, okay? So this is because it has CD4. This CD4 marker here means that this is a T helper cell. All right, so we have interacted with an antigen presenting cell that has MHC2. That means not only MHC2 not only means that is coming from outside, but it also means Lauren's cell, specifically Lauren's cell, is saying, I picked something up from outside. That's what MHC2 says. Lauren's cell picked something up from outside. The T cell receptor says, I see you picked up COVID spike protein. I recognize COVID spike protein. The CD4 marker on the outside says, okay, I see you, T cell receptor. You see that there's COVID spike protein, but I've got to make sure that Lauren's cell is presenting this and saying it's coming from outside. All of this stuff has to happen all together in order to activate. All right, so once we have activated, and we can actually send signals back and forth from each other in order to have this signal happening. Okay, so now we've got this activation going on, and finally, our T helper cell can then differentiate, proliferate. First, we'll put proliferate. I'm writing it, it's just taking a while for it to catch up. What are you talking about? Okay, there it goes. I about lost it. All right, after all that drawing I did. This will first proliferate into a whole bunch of T helper cells. And then these guys will differentiate. I'm just gonna put diff, okay? These guys will differentiate into T helper 1 and T helper 2. And that is not all. Also, T helper memory cells, which are never going to come up again, okay? But you know what a memory cell is. Something that just hangs out after it's been activated once. It's something that once we see it again, can immediately get activated without having to go through this whole BS that we just went through. So T memory cell, as well as a T helper regulatory guy. Again, we're not going to come back to those guys, but they're just going to help keep you from overreacting. We're making sure that you are reacting as much as you're supposed to be. Yes, so after we have activated our T helper cell through this MHC2, help, I have something that came from outside. T cell receptor comes and says, yes, I recognize you're holding out COVID spike protein, and that's the one I recognize. The CD4 verifies Lauren's cell is saying something from outside came over here. We get both of those signals. We activate the T helper cell. It proliferates to a bunch of T helper cells. And then those T helper cells differentiate into these guys here. So we're always going to have, no matter what, all of our lymphocytes will do regulatory, memory, and then these guys here are our effector. This is a T, I promise. Okay, so these are our effector guys. These are the ones that are doing the work. And like we said over here, these are our captains. So next, I'm going to show you what the captains are actually going to do to regulate all the other guys. All right, so this is how everything has to start. This has to happen before any of our other cells can do any other work. Right, so let's look at what happens with an inside infection. All right, inside. All right, so you have an infected self cell. And it's got virus in it. It's going to realize that, and it's going to post up that virus antigen on MHC1, right? Okay, so I already told you that T helper cells interact with MHC2. MHC1 is going to also be recognized by T cells. B cells do not interact with MHC. Only T cells interact with MHC. The T cell receptor recognizes the antigen on MHC1. And this T cell is a cytotoxic T cell. And it's got a different marker on it. It does not have CD4. It has a different marker on it. This one's called CD8. And CD8 only sees MHC1. Okay, so we've got a T cell here with a CD8 marker. So now we've got any of your infected cells that are infected with something inside of them. Any cell that has an infection inside of it saying, help, help, I found this inside of me. Now we've got MHC1 handle holding on to COVID spike protein that came from inside of the cell. And we are holding up that antigen. The T cell receptor sees, hey, I've been waiting my whole life to see COVID spike protein, and I recognize this antigen, the CD8 is going to come on the side and say, yep, I'm glad T cell receptor that you see your specific antigen. I just need to step real quick and make sure that the arm holding this up is Lauren's MHC1 telling me that it came from inside. Once that is happening, do you think that the T cell becomes activated and can start killing things? It doesn't. Because why? Because it needs the signal from Th1 in order to activate it. This is, I can't spell right now, but that's supposed to say activate. This is how we will keep our cytotoxic T cells from just killing anything that they interact with. They have to get that activation signal from the captain in order to do that, okay? Once they've been activated by the captain that also recognized that antigen, now the Th1 helper cell, which was already activated from outside stuff, sends a signal to the cytotoxic T cell that you can start killing things with inside stuff. You cannot, as a cytotoxic T cell, kill self-infected cells without having the captains go ahead. You have to have that. This is such a huge thing that we will remember how we mentioned how your MHC looks different than mine, and that's why my body will kill your cells, right? This is the reason why. This is the only reason why, is that my T cells will see your MHC and initiate this response here. And so this is a devastating, organ destroying process that can happen here. And it causes such devastation if we attack cells too much like this, that it can actually cause you to die from shock if this is going on for too much. So we can't just have our T cells letting themselves activate themselves. They have to have the go ahead from the captain. Unfortunately, that captain is giving the go ahead in an organ transplant, because as far as the captain's concerned, yeah, that really is a bad guy thing. The captain doesn't know, but that is there to help regulate all of that. So inside stuff is regulated by Th1. All right. I'm gonna give you another example. Something that shows the destruction of the cytotoxic T cell response is going to be poison ivy. Anybody in here ever have a poison ivy rash? Okay. It's okay if you haven't, but you at least know what it is, right? All right. Did you know that not everybody gets poison ivy rashes? There are plenty of people in the world that don't get poison ivy rashes. That's not an abnormality. And also, I want to be very clear about this. The oil that is on poison ivy leaves doesn't cause damage. The oil that's on the poison ivy leaves is completely harmless. It doesn't cause problems. What happens is the oil that's on the poison ivy leaves gets taken in by your cells, and your cells go, uh-oh, something's inside of me, and start screaming out, I need an inside response. And then your body attacks your cells that have the oil on them. The oil's not doing anything. Your body is destroying itself because it thinks it has an inside thing going on. And so have you guys noticed also allergies? You have some allergies that you'll just get hives and itchy from, or you'll have other allergies where you have these weird blisters like you get with poison ivy. There's actually two different cellular allergies. You have allergies that are going to be propagated by the antibodies made against things, which we're going to talk about in chapter 17. Or you have allergies that are propagated by your cytotoxic T cells attacking something. So when you have allergies attacking your cytotoxic T cell response, you get what I'm saying? When you have something that your cytotoxic T cells are attacking, it literally destroys your cells. And so that's when you get those weepy, so I'm putting it in my ear because I'm allergic to metal. And so if I buy cheap earrings, I get this crusty, nasty, itchy rash sort of thing from the earrings because I have cytotoxic T cells trying to destroy my cells that have the metal that they took inside of them, causing that damage. But if you get like a mosquito bite, if you get a mosquito bite, you just get a hive that itches, right? That's because your antibodies to mosquito saliva are causing an allergic response from the allergy side of things. So I'm gonna blow your minds with one more thing, which is in third world countries, they don't have allergies. The incidence of allergies, at least, is extremely low because the allergy response is regulated by IgE antibodies, which we'll get to in a moment. But our body needs to train our bodies on how to deal with our anti-parasitic response. IgE is designed for parasites. We don't have parasites in first world countries. So our bodies never got trained on how to properly use that side of our immune system. And so we have allergies because we're too clean. They don't have allergies. Their bodies were trained to properly deal with these antigens. And so they don't have allergies, but they get worms. So I don't know what your trade-off is going to be, but they're actually looking into therapies nowadays that will use worms to treat things like Crohn's disease, which is an allergy to wheat. Or it's a gluten allergy, right? I believe that's what Crohn's disease is. And they're infecting people with hookworms, and their symptoms go away completely. Now, that's going to mean a lifelong infection with hookworms because they're adults and their immune system is already developed to that level. But if we could potentially introduce parasites to children before their immune system goes through that full-on development, then you could potentially eliminate any risk of any allergies at all. So that's impressive. And the medicine is actually looking at putting that to work. All right. Anyways, so coming back to this, we've activated our cytotoxic T cell because our captain right here, our nice happy Th1 captain, went and gave the go-ahead signal from this interaction down here. And so now our T cell will release out these products called granzymes and perforins. Perforins, why? Perforins do what they sound like. They perforate. They cause holes to form in the infected cell. So perforins are going to poke holes in our infected self cell. Then the granzymes, they're going to be able to get in and digest everything that's inside of there. This leads to something called apoptosis, which is basically cell suicide. We'll just word it that way because it's easier. But the cell knows that it's getting destroyed and it'll just die. And so all of these steps had to happen to get to that point, right? We had to see the antigen outside, activate our T helper cell. It had to multiply and turn into the Th1 cell. Then the Th1 cell had to find the cytotoxic T cell interaction with the infected self cell, and then give it the go-ahead to go ahead and poke holes in it and granzyme it and break it down. But when you see this happen in real time, it's like that, as far as when your cytotoxic T cells actually cause the damage. It takes a long time to get your captain. But once that captain is there, activating the stuff downwind is almost instantaneous. And there's videos of a tiny little cytotoxic T cell floating up to like this building size self cell. It comes into contact with it and that cell just goes instantaneously. And it's so impressive. And all of that happens because we're getting this signal from our captain to have the go ahead to do that. So that's our inside response. Let me clean this up a little bit and I'm going to show you our outside response. Clear up a little space here. Maybe if the computer can catch up with what I'm trying to do. Okay. So for our outside response, we've already seen outside with our TH cell, but that didn't get us anywhere in fighting the bad guy. So what actually will happen with the outside stuff, in order to fight the outside bad guys, we're going to need our B cells. And so what happens with the B cell, here's our B cell. We already know what our receptor is like on the outside of the B cell. We've got our nice happy little antibody situation attached to the outside. And this is our receptor, also known as IgD. And that IgD will come into contact with anything floating by in your lymph. Your B cell's hanging out in your lymph node. It's floating by in your lymph. It picks up a COVID virus. It will take that whole thing, receptor inside to its cell, and then put that antigen on MHC2. Because it picked it up from outside. You following? So that's on that outside. And so what is it going to interact with? It's this up here. I'm not going to redraw it, okay? Well, redraw it a little bit. It's kind of going to be a little sideways. This is our T cell receptor on our TH helper cell. And then we're also going to be using CD4 to engage with MHC2. But this won't just be any TH cell, this will be a TH2. And this TH2, because it's been activated and it's seen its antigen before, can go ahead and activate the B cell. Now, this can go two ways. If the B cell, because the B cell can act as an antigen presenting cell as well, the B cell could come in and see this naive guy that's up here that's never been activated before. That B cell could see that guy that's not proliferated, that's not a TH2 yet, and its interaction can cause it to proliferate and differentiate and become a TH2. But then if that happens, if it's never seen it before, it's not a TH2 yet, it has to wait for the TH to become the TH2. First, it's got to proliferate, then it's got to differentiate, and then it's got to re-interact with TH2 in order to get its signal to activate. So TH2 has to activate B. So our B cell here has met an active TH2 cell. It has received its activation signal. So what is the B cell going to do? It's going to proliferate and differentiate into the three Bs, the B memory, the B regulator, and the B effector, which is known as a plasma cell. These are all cells, groups of cells after we've proliferated that we'll differentiate into. The plasma cell is the one that just cranks out antibody. I bet you're still kind of wondering, probably not dying to know, but you're kind of probably in the back of your mind wondering, okay then, but what does an antibody do? What does an antibody do though? No worries. We're going to talk about that soon. Okay, this process. Questions. I know it's a lot. I have spent semesters learning about this stuff. And when I tell you that how I'm teaching you, this isn't even a tiptoe into the ocean that is immunology, but this is the big, big basics here, okay? So to recap, you see something from outside. Your body, you step on a nail. Crap gets inside of your body, neutrophils come and eat it up. They crap out what they eat up. Macrophages come and clean it up. They present on the MHC2 outside handle saying, I found a bacteria antigen outside. I found something outside. The T cell that sees that bacterial antigen reacts with that with its T cell receptor. It says, I recognize bacterial antigen. Its CD4 says, yes, Lauren's cell found something outside. We have the go ahead. The T helper cell then turns into the two different versions, TH1 and TH2. If this was bacteria that was outside, that TH2 will govern your B cell activation by interacting with the antigen presenting from the B cell IgD receptor and putting it onto MHC2 to interact with TH2 to get activated so the B cell can turn into a plasma cell and make antibody. If we have something that's going to live inside of cells, all of the infected cells after this point will be screaming with MHC1, help, help, I found something inside of me. A cytotoxic T cell will come up and say, I recognize that inside antigen. Its CD8 will say, I recognize that Lauren's cell says they found something inside. When that happens, they will also have to receive a signal from TH1 in order to activate our ability to make granzymes and perforins to kill our self-infected cells. That's our summary. Okay, we're moving on. Cool. All right, so the cell-mediated immunities, that's our T cells. I'm going to move through these because these are the words that go along with the pictures that I explained. It's just easier to draw it all out all at once. If you want to watch a review on the T helper response, there's a little video here. This is a summary of T cell activation of our cytotoxic T cells, which are our self-killing cells for inside infections. They produce perforins and granzymes. We don't just use cytotoxic T cells to kill virally infected cells or just pathogens that are inside cells. Our cytotoxic T cells are also going to be destroying cells from other animals and people, but also cancer cells. So your body can actually cry out for help saying that there's cancer going on as well, because that's an abnormal process in the body. And so you might be thinking, yeah, but then why did my uncle have colon cancer if it's his T cells could have controlled that? Or why did my aunt have breast cancer or whatever? Well, your cytotoxic T cells weren't particularly effective at controlling those types of cancers. But I want to tell you guys, there are plenty of other cancers that you would have right now if it weren't for your T cells. In fact, when you see somebody, let's go back to this picture really quick. Oh my God, it's so far back. Okay. If we come back to this picture, and I'm telling you that you cannot activate your cytotoxic T cell response to kill infected cells without Th1. You cannot activate antibody production without Th2. Our Th cells are our helper cells. They have this marker CD4. What do you guys remember what cells HIV infects? CD4 positive T cells. HIV infects CD4 positive T cells. So now do you understand why somebody with an HIV infection has an immunodeficiency? Because they cannot activate their B cells or their cytotoxic T cells. This should be a big picture kind of event here that we're starting to think about, right? Because if you're infected with HIV, you have no captains. That's it. When that response whenever that virus starts taking over and we get to a certain point where your T cell count is low, you don't have enough captains to govern protection in your body. And so that's where we get acquired immunodeficiency syndrome, or AIDS. It's from having an HIV infection infecting your CD positive T cells. In fact, the door handle that the HIV key uses to get in is CD4. So their key unlocks CD4 so they can infect anything with CD4 on its surface. So that's how AIDS comes around from HIV. And that's also why when we start thinking about T cell deficiencies, if you have a patient that has a B cell deficiency or has a problem making immunoglobulin, they're going to be better off than somebody who has a T cell deficiency, because the T cells govern not just the T cells, but also the B cells. The B cells can't act if you don't have the captains. So it's actually pretty important to have that full circle come around so you can understand the importance of the Th cells. They themselves aren't fighting the bad guys, but nobody can fight the bad guys without the captains go ahead. So you eliminate those guys and you've got a problem. People who have HIV have a high incidence of two kinds of cancer, Burkitt's lymphoma and Kaposi's sarcoma. You may have heard of these, but Burkitt's lymphoma, let me tell you where that comes from. 90% or more of adults, once you have reached adulthood, 90% or more of adults have had mono at some point in their life. You may not remember it. I don't remember it, but I have had mono. I've had antibodies against mono in my body, but I never had any symptoms of it. This is actually more common than you think. So little kids that got infected with mono usually don't have any symptoms. It's when you're in your teenage years that you actually have the symptoms of mono. Yes, so we'll talk about that a little bit. Yeah, we'll talk about that a little bit too. So mono is kind of unique. Mono is a herpes virus. So once you've had mono, you have it forever, just like you do any other herpes virus, like chickenpox or herpes simplex virus, right? So once you have mono, it hides out in your lymph nodes. That's where it lives. And so potentially it can come back out at any time. And so you actually have people who have mono and then it's just like chickenpox. Some people that get chickenpox, they've got it once and they may not ever get shingles ever again. But then you have other people that got chickenpox and they're already getting shingles at like age 25 and they're getting it constantly after that. So mono is the same way. You can have recurrence of mono that's already in your body because just your own immune system isn't keeping it at bay. Okay, so you can have mono, and when you've gotten mono, you have it in your body forever, just like herpes. You don't get rid of it. Well, what happens is mono hides out in your DNA and can cause something called Burkitt's lymphoma. So if you've had mono, you are actually pretty likely to develop Burkitt's lymphoma, but your cytotoxic T cells are killing it when it starts to pop up. So it's killing, it might not be very good at fighting the mono, but it might be very good at fighting the Burkitt's lymphoma. And actually your cytotoxic T cells are very good at fighting Burkitt's lymphoma. So all of us might right now in this room be ticking time bombs for Burkitt's lymphoma. Somebody with HIV, they get Burkitt's lymphoma. They don't have the cytotoxic T cell response. They're not getting signals from the captains. So they get cancers that other people don't get because we have our cytotoxic T cells fighting off our cancers. Now tell me that's not cool to think about how that stuff is all going to work in your body. It gets way complicated after that too, but we're going to leave it at these. I know it doesn't feel simple to you. Trust me when I tell you this. Okay. All right. So the B cells. So once we've activated our B cells, and now we can make our plasma cells, can make our antibody, right? And we talked about the structures of our antibody, the FABs, the variable region that interacts with the antigen, and the FCs, the constant region that dictates basically what the antibody can do. All right. So now let's talk about the functions of antibody. All right. Antibody has 6 functions. The first one, coding. It's literally just like if you were a bad guy and I cover you and smother you in blankets, you might have a hard time fighting out from that, right? So we can literally just cover these guys in antibody, and then the bacteria or the viruses just have a hard time doing whatever they're trying to do, adhering to surfaces, multiplying, trying to swap DNA with other guys, whatever it is they do, coding them can stop that. The next one is called opsonization. And we already introduced opsonization when we talked about complement. Opsonization is providing a molecular handle for phagocytes to grab. So this makes it a lot easier for our phagocytes to eat up the bad guys, especially if they have a capsule that makes them slippery and hard to grab onto. So that's opsonization. Antibody can also neutralize. This is essentially blocking docking. So now those spike proteins that act as keys to enter in the doors on our cells, they can't, right? We're literally like putting a big old stop sign, sticking it onto the end of the key so the bad guy can't use the lock picks to get into your house. That's really what we're doing there. So blocking the docking. So this is a little bit more specific kind of blocking. The coding, just coding in general so it can't do things. The neutralization, specifically blocking the docking. The next one is called agglutination. This is causing our bad guys to stick to one another. Just like our fibrin clots and our, we'll cause our platelets to stick together to create, and our red blood cells too, to create our scabs. This is going to cause our bad guys to stick together. So they're going to kind of agglutinate together. So if they're stuck together, if we're gluing all of our bad guys together, they can't get around the house as easily to steal the things and cause damage. Another thing the antibody does that we already talked about, it activates the complement pathway. So of our 3 ways of activating our complement pathway, one of them, the classical pathway, is activated by the presence of antibody. And now we can poke holes using that membrane attack complex. And finally, antibody can act as antitoxin, not binding to the bad guy or interacting directly with the bad guy, but the toxic products that it makes. And a lot of our bad guys, our pathogens, rely on the action of our toxins in order to be able to cause active infections in our bodies. So blocking their toxin can actually block the infection from being successful. Good on those 6 functions of antibodies. Any questions on that? We have 5 classes of immunoglobulins. So all of the antibodies, there's 5 different versions. We learned about 1 so far. This was IG. This isn't even it. The freaking pictures are off-centered from the actual names. It doesn't matter. IgD, and I'm never going to show you the picture, so it doesn't matter. But IgD is the receptor 1. Okay? So that's the one that's on the surface of B cells that can recognize the antigen. If I'm a B cell and my IgD has bound to COVID spike protein, I will suck it in and then put on my MHC, the antigen that was on my IgD, and then on my MHC too, right? Because I got it from outside. My Th2 will then come and activate me so I can turn into a plasma cell and start secreting these other guys, okay? The first one that we make always is IgM. And IgM is this guy right here. It's a pentamer. So this guy we can just crank out immediately. So B cells, once they've been activated and we've got that signal from our Th2 captain, we will proliferate. And then we'll have some of them turn into memory cells, some of them turn into regulator cells, and some of them turn into plasma cells. Those brand new, freshly made plasma cells can crank out IgM immediately, but they're still undergoing major changes in their genetic makeup in order to make better antibodies. And this will be based on signals we're getting from our environment, as well as interacting with other cells. You're welcome that I'm not going to make you learn about that part of it, but those signals in other cells will dictate which other antibodies will switch to. So you always start first with IgM, and then we will switch. So we always, I would say back it up. The pre-show is IgD as the receptor. Then when we start making and secreting antibody, we'll start secreting IgM, and then this will switch to one of these other guys, either IgG, IgA, or the allergy-associated IgE. All right. So it's actually important that we understand these other three guys too. We've already mentioned IgE. IgE is usually involved in parasitic infections. If we don't have proper training going on early on in our development, IgE will bind with things like pet dander or peanuts or shellfish or whatever crap you can be allergic to. You know, cottonwood or whatever you're allergic to, cedar, who knows, pollens a lot of the time. So this is usually used for the parasitic responses, but in first world countries, it's usually associated with allergies. You usually make this in a very small amount, but that reaction obviously can go quite haywire because allergies, we have two kinds of allergies. We call local allergies or systemic allergies. When you have a local allergy, this will lead you typically to like eczema. So eczema, even though we think you can get it all over your body, you're getting it on your skin. You're not getting it in your organs. So eczema is a local allergy response. If you have hives on your skin or you get itchy from being allergic to something and you don't have those weepy wounds going on with them, then that's eczema. Even if it's from being in contact with something, eczema is anytime you have the hives associated with the itchiness. You have the weepiness, like we see with poison ivy, that's called contact dermatitis. And that's the cytotoxic T cell response. That's not IgE. All right. So we've got our IgE associated with parasites or allergies. And it's usually allergies because it's going haywire. It's when your immune system goes wrong. Then we have two other antibodies. IgA is associated with your mucosa. So this guy will get secreted into your actual mucus areas. So in the inside of your lungs or in your GI tract is the biggest ones that we can think of, that IgA will be working in the mucosa to provide antibody protection there. So we call it secretory, not because it's a secret, but because it is secreted. All right. IgD though, IgG is the workhorse. Or you could think of IgG as being for giant. This guy is the big daddy. This is the guy you want in your immune protection. If you want memory that actually protects you, that works so fast that you didn't even know that you walked by somebody that had measles or diphtheria or pertussis, or that you stepped on a nail that had tetanus in it or something like that, you need IgG. IgG is the big daddy giant workhorse. 80% of your antibody response will be IgG once you start making it. But first, you have to make IgM. IgM does not work as well, and it can't do some of those 6 responses like we talked about earlier. It can't do some of them, but it's something to help you get started. But once you start switching to IgG, you are good to go. You are protected, and you're going to clear that infection. So all of the vaccines that we get in our life are designed to try to target production of IgG where we can, because it gives better protection. A titer is a measure of how much antibody you have, and we can actually measure how much IgG you have versus IgM, and that can actually tell us where you are in your infection response. Your primary response is going to have a latent period. This is because first you have to get neutrophils there to eat things up. Then you have to get macrophages there to eat up what the neutrophils left behind. Then the macrophages have to activate the Th cell, and it has to proliferate and then turn into Th2 daddies. Once the Th2 daddies go out, now they can go and activate the B cells to make antibody. That crap takes a while. It takes a minute, and we're talking several days. It's why whenever you get a new infection, you're sick for a few days. Okay? Waiting on your antibodies to come about. Your innate system is working until these guys come. Once you start making antibody, you're usually going to clear it very, very quickly. So your first exposure to something is latent because we got to wait for those things to come up. That's why when you get the flu, you're going to be sick for a week. You're literally waiting on your antibody response to just happen, and it takes that long. Do you have a question, Ida? So the medicine helps you feel better, but it might also dampen your immune response. So that's a give and a take, right? So if you have, and so it depends on what medicine too. You mean like Tamiflu or something like that? So right. So if you take like acetaminophen or something like that, acetaminophen will lower your fever response, and your fever, until you can get your antibodies made, fever is all you got, really. Your fever and your neutrophils, right? And so your fever helps your neutrophils act faster and better. Your fever also speeds up your B cell response. So dampening a fever can slow this process down. But if you're in a lot of pain, don't let me stop you from taking the medicine, right? Your immune system will eventually get there. So this is where we talk about like weighing the pros and cons. If you're in a tolerable amount of pain, maybe let your fever help you out. But if it's like a lot of pain, you need some help, take the acetaminophen or the ibuprofen or whatever. And so that's a problem. So we learn about steroids. Steroids dampen your immune response overall, and steroids can actually make you more likely to be able to get infections because it's literally just silencing your immune system. So it'll treat symptoms. So when you get like a sinus infection, we often get prescribed steroids. And so it'll be fine probably, because probably your immune system will still be able to overcome it. But if you have bigger infections or something that's not being controlled well by your immune system, it can actually make things worse, especially if you're on steroids over a long period of time. So like I said, steroids are fine for infections if they're coming along with something to help out your immune system, like an antibiotic, you know what I mean? So depending on the drug and over-the-counter stuff, just I would assess your situation and make your choice. But now you can make it in an educated way, right? You know a little bit more about why you have fevers and things. Okay, so your primary response is delayed, and it could potentially be more delayed if we do take medications to treat our symptoms, potentially. But that latent period is the amount of time that we're waiting on the activation of your B cells and their proliferation and differentiation to be able to make antibody. The first antibody that they are going to be making is IgM. It is not particularly effective, but then later on, you'll start seeing production of IgG, and that's better. But then this is maybe years and years. So for me, right, I had chickenpox when I was 7. And so now if I see somebody that has chickenpox again after that, every time that I encounter somebody that has the chickenpox, I get my secondary response. And it's so quick and fast because we're making IgG, our big daddy workhorse, that's going to be our giant, fighting things off immediately. We don't have to wait for a latent period of a week. We can immediately start cranking that stuff off because our memory cells can just start making it. We don't have to wait on proliferation or anything like that. So this is why we have vaccines, and why vaccines work as well as they do is because we are eliminating the latent period, and there's other things involved in vaccines that help them work better as well. When you're exposed to something naturally versus exposed to something from like a vaccine or something, natural exposure to something can lead to antibody development that can be protective. That's natural immunity. Artificial immunity is whenever I give you a vaccine, I've introduced this thing artificially, and now you have antibodies because of that artificial interaction. Your immunity can be active or it can be passive. Active immunity means you make your own antibodies. Passive immunity means I gave you someone else's. Passive immunity is a little bit strange, right? We don't think about this a lot as something that happens naturally, but it does. So when we breastfeed our babies, there are antibodies in the colostrum. And so all of the antibodies that the mama makes for her own protection get passed into the baby, and the baby actually gets protection from the mama's antibodies. The baby isn't making those antibodies, the mama is. And so this is natural passive immunity. Artificial passive immunity is something we think of a little bit more in the medical field whenever we go get immune globulin infusions. So you probably don't need them, but you may have patients or heard of patients that have needed immune globulin infusions. These people don't make enough antibodies on their own. And so we take the plasma from other healthy people and just put their plasma in the sick people. And then for the meantime, they're circulating those antibodies around. It gives them random immunity from whoever their donors were. Another thing that people sometimes don't think of, Humira. So we talked about Humira before. It's adalimumab. MAB means monoclonal antibody. So when you get Humira injections or infusions, you're just putting antibody in that works against the molecules in your inflammatory cycle. But it's an antibody that we're giving you passively or artificially, because it's given medically. Okay, so that's another example of an artificial passive immunity. Okay, and for our vaccines to work, this is the end of it, the vaccine part. We have vaccines that work from whole organisms and vaccines that just work from the antigens. If you're getting a whole organism vaccine, these organisms could be killed. This is an example of this would be the flu vaccine. Flu virus is dead if you're getting the injections anyways. So if you're getting the nasal spray because you're allergic to eggs, that's a different story. But if you're getting the injections, which is most of the people, those are dead viruses. They literally cannot cause the flu. They can't. It's like impossible. Like keep dreaming, okay? It's like zombies. They aren't real, okay? So it's not happening. So we'll talk about why people get sick from flu vaccines in a minute, all right? The other one are live attenuated viruses. These guys are actual viruses that do multiply in your body and they're intended to, but we've changed them in a way that they shouldn't be causing disease. Polio vaccine is an example of this. Polio vaccine is a live attenuated virus. What with polio vaccine, there are some very, very rare circumstances where they can revert back to the pathogenic form. So that's the danger with live attenuated vaccines. We don't see it happen very often, and it almost never, I don't know if there's ever even been a case with the MMR vaccine of that happening, but it works in a similar way. Then we have other whole virus vaccines that is like one kind of virus, like a cold virus that we can hollow out and then put in like the spike protein genes for COVID. That's how the Johnson & Johnson vaccine works. So if you got Johnson & Johnson, then you got an adenovirus that we put COVID genes into, and instead of having adenovirus spikes, it has COVID spikes. And then that's how it elicited the immune response that way. Then our antigen-based vaccines, we're either just literally just injecting you with the antigen, like the spike protein itself. That's how the hepatitis B vaccine works. Or the COVID mRNA vaccines, this is Moderna and Pfizer. Moderna and Pfizer, we are putting a vesicle with mRNA in it. We inject it into your arm, and then your arm starts expressing the mRNA. There's a problem with that, and it is the reason why the mRNA vaccines don't give protection the same way that like MMR, like measles, mumps, rubella vaccines do. We have already talked many, many times about how effective MMR vaccines are and how you can see somebody with measles and never have any symptoms. But yet with COVID, you can get the COVID vaccine and still get COVID. So what's up with that? There's a reason for that, okay? And then we can also have antigens that we link to other things in order to make it easier to react to them. Our memory cells are not eternal. They are not immortal. So your memory cells need to be constantly stimulated in order to make sure they're staying around. Some of them last longer than others. And so boosters vary based on the antigen, as well as how the vaccine was delivered and et cetera. So booster shots are there to make sure your memory stays around. We can administer vaccines based on essentially how that virus, we want your body to respond to that virus. So some vaccines are given subcutaneously, right under the skin. Some are given into the muscle. That's a pretty common one. It helps with the dissemination of the antigen. Or intradermal. Intradermal means into the skin. That's how they give the smallpox vaccine, which we don't get them anymore. But they will excoriate your skin and literally scratch into it. And that whole process of the scabbing and everything helps with the actual immune property of that particular virus. Then we also have oral and nasal vaccines. So these are not given very often. An adjuvant is anything that is added to a vaccine to help boost the response. So a lot of our vaccines, like the MMR vaccine, we will put in molecules and chemicals that help trigger IgG production. We don't want your body creating a response for inside stuff. We want your body making IgG, which is an outside stuff. We don't ever want measles, mumps, or rubella to get inside. So we need a very strong outside response. And so we put things in those vaccines to trigger the outside response, to trigger IgG production. And it works extremely well. That's what adjuvants are in there for, okay? When you get the COVID vaccine, coming back to that, and this is me coming with this full circle, okay? When you get the mRNA vaccine, we have to wait for that mRNA to be translated by your freaking ribosomes, and that takes time. The signals that are involved in giving the cues for the IgG production, they don't stick around long enough to elicit IgG. So instead of getting IgG response, you get inside response, which is your cytotoxic T cell response. You still get memory from that, but it just doesn't work as fast. So you're still gonna get sick with COVID. We're just waiting on the cytotoxic T cell stuff to start engaging. It's like if for those of you that have had poison ivy, then you know this, or if you have jewelry allergies like I do, where you'll wear the earrings and a couple days later, you have the reaction. With poison ivy, you contact the poison ivy, a couple days later, you have the reaction. That's because it takes a couple days for your cytotoxic T cell memory cells to kick back into gear, because they can't just crank out antibody. They have to remultiply up the cells, and the cells are doing the work. And there's a delay in that. We call that delayed type immunity because of that. And that's what's going on in COVID as well. You're going to get sick, but eventually your cytotoxic T cells will fight it off. And then you'll also have a much lesser illness. And it's the whole reason why you and I are here in this room right now without masks on, is because those vaccines work and that response, okay? So those are adjuvants. Last thing, side effects. If you get a vaccine and you have side effects, let's say you get a vaccine and your arm hurts. Well, duh, we just shoved some stuff in your arm, right? If I stab you with a knife in your arm, that's gonna hurt too. Okay, so that's gonna hurt, duh. But when you get flu-like symptoms from a vaccine, that is your immune system building up immunity. Not everybody does that to every vaccine, but based on your immune system might have immune responses that you'll feel, like fever, like soreness, like fatigue. These are all your immune system. In fact, 99% of all illness that you ever get, the bad feelings that you have of your signs and symptoms, is usually actually propagated by your immune response, not by the actual infection. So that's something to remember as well. There is a risk of anaphylaxis if you're allergic to something in the vaccine. It's pretty low. In the case for COVID vaccine, anaphylaxis risk was 5 cases out of every million. So it's still something to be concerned about, but that would be an allergy to something in the vaccine, not to the COVID itself, but maybe to the fact that it was developed with certain molecules in it. All right, vaccine misconception. Do I need to tell you again that there is no link between the MMR vaccine and autism? There never has been. The only links that were ever posted about that were falsified evidence by a guy who has lost his medical license and literally was paid to make up those results. All of those results have been retracted, and all of those papers have been removed. But he is still allowed to publish, of course, YouTube videos and, convince, celebrities and things like that to endorse his teachings so that he can continue to make a living. But there's absolutely no evidence that links the MMR vaccine with autism. I am not saying that vaccines can't cause injury. Of course they can, but MMR being associated with autism, that is not the case. Probably actually more likely that autism is more of a societal type of effect of dealing with. Have you guys heard this before? There's this talk that I saw this guy give once. It was a TED talk or something. He's talking about how human minds are not meant to know what's going on in the world. Humans were meant to live in groups of maybe 150 people, and you're meant to know what's going on with your group. And that being exposed to society and knowing all of society's problems and having to worry about what your president is doing and what the nation is doing and how there's fires in LA and all of these things that are going on in the world, our minds were literally not adapted to deal with all of this trauma all at once. No wonder that we have brain effects from this, leading to mental illness and other things that are causing our brains to not be able to function the way that they were designed to do, what we call neurotypical. No wonder we have so many neurospicy, quote unquote, effects out there whenever our brains were not designed to deal with that. So that's probably more likely explanation for some of these incidences. Not all of them, of course, but some of them banned what we can say with MMR for sure. So if you have any questions about that.
Autism is genetic.
Yes, so it can be genetic related too.
Some like when my doctor told my sister and my mom to go with autism that when she got the MMR shot when she was young, it like I don't know reacts to autism and it just like
There's no evidence of that.
it was like saying I don't know it was so weird. So I didn't know that it
So-
was like it.
So there just isn't any evidence of that, and people have latched onto the societal jargon about it. There's no actual scientific evidence ever. Like, and we're not just saying that to cover our asses because vaccines are good. We're like literally there isn't any evidence of it. And people have heard that it works that way, and so this misinformation just gets spread even amongst people in certain medical communities that aren't actually educated about the evidence behind it. Obviously, we have an increase in the incidence of autism. Yes, I don't think anybody's going to disagree with that. You guys probably have heard about this. When we talk about like mental illnesses like bipolar disorder or chronic depression or things like this,
That's all genetic.
that they are genetically, you have a genetic predisposition. But just because you have the genetic predisposition does not ergo mean that you're going to have bipolar disorder or autism. That it is usually a combination between genetic factors and environmental factors. And so that's usually what's going on with a lot of these things. There's genetic aspects to it, but just because you have the genes that make you likely to have breast cancer, even like the MTHFR genes, you have the genes that make it likely you might get breast cancer, but it doesn't guarantee you will get breast cancer. These other environmental factors add into that and increase the likelihood of breast cancer, bipolar disorder, autism, and all these other things. So it's probably more likely, like a lot of things in life, more likely a combination of both genetics and environmental factors. It's not the measles vaccine stuff that probably that is linked to it, but I can bet you it would have been something from around the same timeline.
What's funny is my
Yeah.
mom, she doesn't believe that. She's like, there's no way that MMR
Yeah.
that like gives you autism. So she